Alright, so last time we talked about the structural component of the plasma membrane. Now we're going to actually go into quite a bit of detail about what, in my opinion, and certainly the opinion of Year 11 biology, is the most important function of the plasma membrane, and that's talking about transporting molecules across the plasma membrane. Alright, so here we've got a plasma membrane of this paramecium, and the plasma membrane, it's semi-permeable, that's what we call it, and here the plasma membrane is putting passive into passive transport. And here he says, you want to pass through? Oh sure, go right ahead, that's just fine with me. That's not passive transport, it's something else. Pause the video here and have a go at the 3 quick questions. Alright, so hopefully in these 3 quick questions you saw that question 1 said, choose one organelle present in the eukaryotic cell, describe where it would be found, what is its function, and produce a labeled drawing of the organelle. All right. A good example of this might be the mitochondria, which looks something like this. Beautiful little mitochondria here. I'm sure yours look a lot better. So we have the mitochondria, and function is to break down glucose into ATP. I'd say the function is a little bit more than that, but hey, that's pretty good. You can have lots and lots of different responses for this. You can talk about the Golgi, you can talk about the nucleus, you can talk about the endoplasmic reticulum, lots and lots of. Let's compare and contrast carrier versus channel proteins. This is more of the focus of what we're going to talk about today. But carrier proteins, for example, so first of all, they're both membrane embedded proteins used in transport, right? And they're going to pretty much transport stuff through the membrane, all right? Carrier proteins are specific. Channel proteins are pores, all right? They're just openings in the membrane. Whoops. And then lastly, the question last set says, Mr. Mello wanted to observe a bacterial cell which were infected by a virus. What kind of microscope would you recommend he use? Hopefully you would certainly recommend that I was using a electron microscope. If I'm going to look at a virus, it's going to have to be a pretty powerful electron microscope. Bacterial cells themselves are actually better to be seen through an electron microscope, because they look like tiny little dots, pretty much. Alright, so in the study design, it's the last stop point of crossing the plasma membrane. We're talking about modes of transport of soluble substances across the plasma membrane, including through the process of simple diffusion, facilitated diffusion, osmosis, and active transport. Let's do it. So, remember that the plasma membrane has this hydrophobic and hydrophilic sections. All right, we've got the hydrophilic heads, which are on the outside of the membrane. The reason they're on the outside is because both the outside of the cell and the inside of the cell is water. They're made up of mostly water, right? Whether it's the cytoplasm or whatever surface that the cell is in, all right? And then on the inside, we've got these hydrophobic fatty acid tails, okay? And they're sort of like trying to get away from that water, all right? That water generally, it's not constantly flowing through the membrane, so this is an area where we don't really have water. Okay, and this is going to be extremely important in terms of what gets through the plasma membrane. All right, the first process of diffusion, all right, the first process of that actually getting through the plasma membrane is called simple diffusion. And simple diffusion means that it's using a passive movement system. All right, passive, we mean no energy, or if you're a biologist, we're saying no ATP. P required, all right? And when we're talking about simple diffusion, we're talking about very small hydrophobic substances. So, for example, oxygen and carbon dioxide molecules, right? And these are both going to travel through the phospholipid bilayer from an area of very high concentration to an area of lower concentration, right? And this isn't going to require energy, like we said. So, for example, here we've got a lipid bilayer, okay? And we've got this certain molecule, which might be, for example, an oxygen molecule, all right? Or it might be a small lipid, a small fat. So two different hydrophobic substances there, okay? Lots and lots on the outside. So we're going to get this movement in this direction until we're getting equal concentration. All right, this is what we call that it's along the concentration gradient, all right, where the movement is going from that high to low, and we're looking at stabilizing everything. Okay, we want equal concentration on both sides, if possible. Oops. All right, a special kind of simple diffusion is called osmosis, and osmosis is fusion of water, right? So, although water is hydrophilic, obviously water dissolves inside water, right? It's so small that it can actually squeeze between the phospholipids in the plasma membrane and actually get through the plasma membrane, okay? So here you can see we've got this substance, these dots, and it's too big to get through the plasma membrane, all right? Even though we've got more concentration on this side than this side, okay? So what's going to happen, this water that this molecule is inside is actually going to move across the plasma membrane, all right, across this semi-permeable membrane, so that we get sort of this happening, because now you can see that as much as though we have less water on this side and more water on this side, the concentration of whatever this molecule is even on both sides, okay? So we've got an equal concentration here, okay? Water is always going to move from a region of high water concentration, so we have a low solute concentration. So for example, on this side here, we've got a high concentration of water, but a low concentration of solute, to a region of low water concentration. So for example, here, or a high solute concentration, okay? And this is known as osmotic flow, okay? It's really important to note that water is actually still, so we haven't got a stopping of movement here, all right? But it's just moving constantly from both sides. So in this case here, we have more movement this way. We've got a tiny little bit of water movement back as well. All right, there's a really big effect on cellular environments of this, okay? For example, here, in normal situations, what we call an isotonic solution, there's an equal concentration of water outside and in. So there's an equal amount of movement of water outside and into a cell, all right? This is really good for animal cells. This is what we want in an animal cell. That's normal. In plant cells, we actually call this a flaccid cell because it actually doesn't have the turgor, so that it doesn't have that strength that we need. So plant cells actually want to be in what we call a hypotonic solution. Hypo meaning less than, so the outside has a smaller concentration than inside. What's going to happen here is the water is going to mostly move into the cell with tiny amounts of water moving out, okay? In an animal cell, we're going to get lysis, so a break, all right? In a plant cell, we're going to get turgid, all right, or turgid, and this is a normal plant cell. Hypertonic solutions, on the other hand, is the other way around. We've got really high concentration of waters on the outside and low concentration on the inside. So we're going to get mostly water moving out of the cell. Same here with the plant, and this is what we call a plasmolysed plant cell or a shriveled animal cell, okay? Facilitated transport's another type of transport that we have, right? So when we're talking about a hydrophilic substance such as an ion, so sodium or potassium, and very large molecules like glucose, for example, they can't simply pass through the plasma membrane. Okay, they're very large, all right, or they're hydrophilic, so they're water soluble, so they can't get through that middle section, which is hydrophobic in the plasma membrane. Okay, so what they actually do is they use these special proteins, for example, this carrier protein, right, to actually get through the plasma membrane from a region of high concentration again to a region of low concentration. So see here we've got lots of concentration of whatever molecule this might be, maybe glucose, and so the glucose is moving through this carrier protein into the cell. Okay? The last kind of movement is called active transport. Okay? So whilst facilitated transport is still talking about no ATP, no energy, active transport, here we have energy required. So energy or ATP required, okay? Because there's situations in which cells actually have to carry molecules from regions of low concentration to regions of high concentration. So for example here, if let's say this cell is a muscle cell, it's going to need lots and lots of glucose, and this is a glucose molecule. Even though there's very little glucose outside the cell, it's going to use this carrier protein to move the glucose into the cell. How does it do that? These carrier proteins attract this glucose molecule, and then using ATP, they pull the glucose molecule into the cell. Okay? And that's simply how it works. A really cool example of this is the sodium and potassium pump. Right? Those of you that do psychology, this is a really important mechanism that your neurons have, right? And that's how the actual action potential actually moves down that neuron, right? What happens is we have a really high concentration of potassium outside the cell and a high concentration of sodium inside the cell, okay? But what the cell does is it moves that potassium into the cell and the sodium out of the cell, right, against their concentration gradients, okay? And then once this happens, right, the action potential can then move down the cell and we get an equilibrium happening back. So the potassium moves back out of the cell and the sodium moves back into the cell. All right. As you can see, Drake here, he's showing us the sodium potassium pump, which is an example of active transport. All right. And a really important example for biology.